Welcome to the deep dive. We take your source material, complex topics, and really unpack them piece by piece. And today, you brought something really fascinating, fundamental even, to how medicine works. It's all about how our own genes affect the drugs we take.
Absolutely. Yeah, we're diving deep into pharmacogenomics today. Specifically, we're looking at chapter 68 from this pharmacology text, which is, packed with detail. And it's a field that's really rapidly changing how we think about medicine, that profound connection between our DNA and how we respond to drugs.
Right, so the mission here isn't just a quick skim. We're really aiming to go properly deep into this chapter, pull out all the key insights, understand the mechanisms, the implications, basically equip you with a really detailed understanding of how genetics fits into this whole picture of drug therapy, like getting under the hood, guided by this material.
Exactly. We'll unpack the core principles, how genetic variation actually causes differences in drug action. We'll look at specific genetic variants, the ones with big clinical consequences, and explore how this knowledge is actually starting to be used in clinics right now, and even how it's speeding up the development of new medicines. You'll definitely see drug response in a new light after this.
Okay, let's unpack this then. So to really get our heads around how genetics plays a role, how drugs work differently for different people, where do we even start? The material mentions this shift from pharmacogenetics to pharmacogenomics. What's the difference there? Is it just semantics?
That's a great starting point. It's not just semantics, it's really about scope and, well, technological progress. Pharmacogenetics, that's the older term, it historically focused more on how, say, a single genetic difference, often a pretty rare one, how that single variant in one person or maybe within a family could lead to a really unusual, sometimes quite dramatic reaction to a drug. Think of those early discoveries, maybe mid-20th century.
Like finding one specific genetic reason why someone had a really bad side effect.
Precisely. Often a single gene causing an outlier response. It was more focused on the individual, often reactive, like, why did this one person react so badly? Pharmacogenomics, though, it broadens the lens considerably. It looks at the impact of multiple genetic variations, often common ones, all across the genome, and how they interact across whole populations.
Okay, so multiple genes, common variations, looking at everybody.
Exactly. How does the combination of many variants influence the variability we see in drug response across large groups? Why do some people respond brilliantly, others barely at all? Others get side effects, even on the same dose. It's more systemic, looking at the whole genetic landscape influencing drug response. It's less about the single rare event and more about explaining the common spectrum of responses.
Got it. A wider net, looking at the whole system, enabled by newer tech, I guess.
Definitely. Genome sequencing and analysis tools made it possible. Now, how do researchers figure out genetics is involved at all? The source mentions, traditional Mendelian genetics versus other ways.
Right, Mendelian genetics, like Gregor Mendel and his Ps, tracking traits through families.
Exactly that idea. You look for clear patterns of inheritance for a specific trait, a phenotype, across related family members, trying to link it back to a single gene's inheritance pattern.
Like tracking eye color or certain inherited diseases down a family tree.
Precisely. But applying that directly to drug responses is, well, tricky. Drug response phenotypes how much pain relief you get, how nauseous you feel, how much your blood pressure drops. They're often hard to measure precisely and consistently across a whole family.
Yeah, I can see that. It's not as clear-cut as blue eyes versus brown eyes.
Yeah.
And you can't just give everyone in the family a drug for an experiment, ethically speaking.
Exactly. The practicalities and ethics make those classic family studies very difficult for drug response. So researchers often have to rely on other approaches, usually non-family based ones. Twin studies can give you a good hint. If identical twins who share nearly 100% of their genes have much more similar drug-handling traits, how they absorb, metabolize, excrete a drug compared to fraternal twins who only share about 50%.
Like regular siblings.
Right. If the identical twins are much more alike in their drug response, that strongly points towards a significant genetic component driving that similarity, rather than just shared environment.
Makes sense. A way to start separating genes from lifestyle or environment.
It helps. But the heavy lifting and pinpointing the specific DNA variants usually comes from studies in large groups of unrelated people. And the material highlights two main strategies for that: candidate gene studies and genome-wide association studies, or GDOLAs.
Okay. Candidate gene versus GWAS. Can you break those down a bit more for us? How do they differ?
Sure. Candidate gene studies are, well, they're hypothesis-driven. You start with what you already know about the drug's biology. Let's say you know a drug is mostly broken down by a specific liver enzyme, say CYP2C19. Your hypothesis is that variations in the gene that codes for CYP2C19 are likely candidates for influencing how quickly that drug gets processed. So you focus your search specifically on that gene, or maybe a few related genes.
So you have a suspect list based on the drug's known mechanism.
Yeah.
Let's look at the gene for receptor X, because the drug hits receptor X.
Exactly. You have a biologically informed guess. GWS, on the other hand, is much broader, more unbiased or hypothesis generating. It doesn't start with a specific suspect gene. Instead, it scans genetic variations, typically hundreds of thousands or millions of SNPs across the entire genome in a large group of people. You compare the frequency of these variants in people who respond well to a drug versus those who respond poorly, or those who get a side effect versus those who don't.
Casting a really wide net.
A very wide net. The goal is to see which variants are statistically associated with a particular drug response, even if you didn't initially suspect that gene region was involved.
That sounds incredibly powerful for finding unexpected links. But, like you said, scanning millions of spots, you're bound to find some associations just by chance, right? False positives.
That is a critical point, and the material really hammers this home. GWAS is great at finding statistical associations, but association doesn't automatically equal causation. You do run a high risk of finding spurious associations just due to the sheer number of tests.
So finding something in one GWAS study isn't proof.
Not at all. That's why replication is absolutely fundamental. Any finding, whether from a candidate gene study or especially from a GWAS, must be replicated in independent groups of people using different study designs if possible before it's accepted as a genuine, valid finding. That independent confirmation is crucial for building confidence that the association is real and potentially clinically meaningful.
Okay, so find it once. That's interesting. Confirm it independently, multiple times ideally. Then you can start thinking about clinical relevance. Replication is key. Got it. Now let's get down to the nitty-gritty. The types of genetic variations themselves. What kind of changes in the DNA actually influence drug response? Table 68.1 in our source lists several.
Right. The most common type of variation by far is the single nucleotide polymorphism, or SNP. You'll also see it called SNV, single nucleotide variant. It's the simplest kind, just a change in one single DNA base, one letter at a specific spot in the genome.
A single typo in the DNA code.
Pretty much. And when we're talking about pharmacogens, the genes affecting drugs, the SNPs that often have the biggest impact are non-synonymous SNPs.
Non-synonymous meaning?
Meaning the single base change occurs within the part of the gene that codes for protein, and it actually changes the resulting amino acid in the protein sequence.
Ah, so it changes one of the building blocks of the final protein machine.
Exactly. And if you change an amino acid, especially in a functionally important part of the protein, like the active site of an enzyme or the binding site of a receptor, that can alter the protein's structure, stability, or how well it works. It might make an enzyme slower, faster, less stable, or maybe not work at all.
Does the material give a concrete example of a non-synonymous SNP?
Yes, it points to CYP2C902. This involves a specific change where an arginine amino acid is swapped for a cysteine at position 144 in the CYP2C9 enzyme protein. That single amino acid change leads to a measurable reduction in the enzyme's function. So drugs relying on CYP2C9 won't be metabolized as efficiently if you carry that variant.
Okay. What other kinds of DNA changes are important besides just single letter swaps?
We also see small insertions or deletions in bubbles, they're sometimes called. This is where one or a few DNA bases are either added into or removed from the sequence.
What effect does that have?
Well, if an indel happens within the protein coding region and the number of bases inserted or deleted isn't a multiple of 3, it can cause a frame shift. This scrambles the entire downstream amino acid sequence, usually leading to a completely non-functional protein, often cut short prematurely. But indels, and sometimes even SNPs, can also be important if they occur in regions that control how the gene's RNA message is processed, particularly splicing. If splicing signals are disrupted, crucial parts of the protein might get left out or incorrect parts added in, again leading to a non-functional protein. The example given is UGT1A128, which involves an insertion, but interestingly, it's in the promoter region.
The promoter, that's the control switch for the gene, right? Not the part coding the protein itself.
Exactly. The promoter region controls when, where, and how much a gene is turned on or expressed. Variations in the promoter, like insertions, deletions, or SNPs, can significantly change the amount of protein that gets made. If a promoter variant reduces gene expression, you'll have less enzyme or transporter protein around.
And that affects drug response, too.
Absolutely. VKORC1/2, mentioned in the table, is a classic example of a promoter variant. It reduces the amount of VKORC1 protein produced that's the target for warfarin. Less target protein makes you more sensitive to warfarin.
Okay, so changes in coding regions change the protein's function, changes in regulatory regions change the protein's amount. What about these synonymous variants the source mentions? Synonymous means the DNA change doesn't alter the amino acid sequence, right? So why would they matter?
You're right, that's the definition. It seems counterintuitive. Because the genetic code is redundant, some DNA-based changes don't actually change the amino acid specified. You'd think these silent mutations wouldn't matter. But the material notes that accumulating evidence suggests even these can subtly alter pharmacogen function. The leading idea is that changing the DNA sequence, even without changing the amino acid, can change which specific codon, 3-base code, is used. Different codons for the same amino acid can be read by the cell's machinery at different speeds.
Different speeds.
Yeah, altering the rate of translation, how fast the RNA is read to make protein. This change in speed might affect how the protein folds up into its correct 3D shape as it's being made. If it folds incorrectly, even slightly, it might not function properly. The example given is ABCB1 RS1045642, a synonymous SNP that might affect the stability of the mRNA or the folding of the resulting P-glycoprotein transporter.
Wow, that's subtle. The whole process matters, not just the final amino acid sequence. Okay, what about bigger changes, like whole chunks of DNA?
Yes, copy number variation, or CNV, is also really important. This isn't just small changes. It's where whole genes or large segments of DNA are either deleted entirely or duplicated multiple times.
So you could be missing a gene altogether, or have extra copies.
Exactly. A gene deletion, like the CYP2D65 allele, means that copy of the gene simply isn't there, so it produces zero functional protein. On the flip side, you can have gene duplication, like CYP2D61XN. The XN means multiple copies. Someone might inherit 3, 4, 5, or even more copies of the functional CYP2D6 gene on one chromosome.
And having extra copies would mean way more enzyme activity.
Precisely. More gene copies generally lead to more protein production, resulting in significantly higher enzyme activity. This is the basis for the ultra-rapid metabolizer phenotype we'll talk about. The material also points out that detecting all these different types of variants, SNPs, indels, CNVs, can be challenging. A single testing method, like a standard SNP chip, might miss deletions or duplications. You often need a combination of techniques for a complete picture, especially for complex genes like CYP2D6 or the highly variable HLA genes involved in immune response.
Right. And looking back at table 68-1, the frequency differences across populations just jump out. European, African, East Asian ancestries often have very different chances of carrying these specific variants.
This is a massively important point with real clinical consequences. The allele frequencies for key pharmacogenetic variants can vary dramatically depending on someone's genetic ancestry.
So one group might have a much higher prevalence of, say, a poor metabolizer allele for a certain drug.
Exactly, which means, as a group, they might be at higher risk for toxicity with that drug at standard doses, or conversely might need higher doses if it's a variant causing ultra-rapid metabolism. The VKRRC1 promoter variant affecting warfarin dose is a perfect example again. Super common in East Asians, over 90% carry it, fairly common in Europeans, around 40%, but much less common in Africans, around 10%.
So considering genetic ancestry can be a really important piece of the puzzle when thinking about potential drug responses for an individual. It underscores the need for diversity in genetic research too, doesn't it?
Absolutely critical. We need data from all populations to understand these differences and ensure equitable application of pharmacogenomics.
Okay, let's dive into how these genetic differences specifically impact pharmacokinetics. That's the ADME part, right? Absorption, distribution, metabolism, excretion. Basically what the body does to the drug.
Exactly. And the source material states clearly that clinically important variants have been found in genes influencing all these steps, but especially metabolism and transport. A big clue that a single gene is playing a major role in a drug's pharmacokinetics is when you look at drug concentrations in a population and see distinct groups.
Not just a smooth bell curve, but like separate peaks.
Precisely. If you measure drug levels after a standard dose and find, say, one group with very high levels, another group with very low levels, and a main group in the middle, that strongly suggests a single gene polymorphism with a large effect, often related to how the drug is metabolized or eliminated.
And figure 68-1 shows this classic picture for CYP2D6 activity, doesn't it?
Yeah.
You see those clear bumps representing different metabolizer groups.
Yes, that figure is iconic in pharmacogenomics. It illustrates these distinct metabolizer phenotypes based on enzyme activity. You have the poor metabolizers, PMs, at one end, with very low or essentially no functional CYP2D6 activity, they form a distinct group because they usually inherited two non-functional alleles.
So their enzyme for that pathway is basically switched off.
Or severely impaired, yes. Then you have the intermediate metabolizers, IMs. Their activity is somewhere between PMs and the main group. They might have one working allele and one non-working one, or maybe two alleles that both have reduced function. The biggest group is usually the normal metabolizers, also called extensive metabolizers, EMs. They have what's considered the normal level of enzyme activity for the population, typically having two fully functional alleles.
But the figure also shows ultra-rapid metabolizers, UMs, at the other extreme. Where do they come from?
EMs, as we touched on with CNVs, often have gene duplications. They've inherited more than two functional copies of the CYP2D6 gene, maybe 3, 4, or even more copies. This leads to significantly higher enzyme production and much faster metabolism than EMs.
Okay, so genetics slots people into these bins, PM, IM, EM, based on their inherited enzyme capacity. The source mentions assessing the diplotype, that the specific pair of alleles someone has can get complicated. Why is that?
It gets complicated because for many of these genes, like CYP2D6, there isn't just one bad allele or one good allele. There can be dozens, even hundreds of different known variants, each potentially having a slightly different impact on function. Some cause complete loss, some partial reduction, some might even increase activity slightly, though that's rarer for single SNPs. And remember, you get one allele from your mother and one from your father. So your diplotype is the specific combination of those two. To predict your overall metabolizer status, PM, IM, EM, UM, you need to know which specific variants are on each of your two alleles and what the functional consequence of that combination is. It requires referencing complex databases and activity scoring systems. It's not always straightforward.
Right. And figure 68-1b is super helpful because it shows the actual consequence of being in these different metabolizer groups in terms of drug levels in the body over time.
Absolutely. It perfectly illustrates the gene dose effect on drug exposure. After a single dose, UMs, with their high enzyme activity, clear the drug the fastest. So they have the lowest peak concentration and the lowest overall exposure, the smallest area, under the curve, or AUC. PMs, at the other end, clear the drug slowest because their enzyme isn't working well. They end up with the highest peak concentrations, the longest drug half-life, and the largest AUC, meaning the most drug exposure from the same dose. EMs and IMs fall in between.
And this difference gets magnified with the repeated dosing, like taking a pill every day.
Exactly. That's where it often becomes most clinically relevant. With chronic therapy, these differences in clearance rate lead to very different steady-state concentrations. PMs will accumulate the drug to much, much higher levels than EMs, potentially reaching toxic concentrations. IMs, conversely, might clear the drug so fast that their steady-state levels never even reach the minimum effective concentration, meaning the drug might not work for them at standard doses. The time it takes to even reach that steady state is also much longer for PMs. So the genotype directly dictates drug exposure over time.
Let's walk through some of the specific examples the source provides, starting with the big family of cytochrome P450 enzymes, the CYPs. CYP3A comes up first.
Right. The CYP3A subfamily, especially CYP3A4 and CYP3A5, are workhorses. They metabolize the largest number of drugs currently on the market. Now, CYP3A4 activity varies hugely between people, like a 10-fold difference or more.
10-fold, wow.
Yeah, massive variability. But interestingly, variants within the actual coding sequence of the CYP3A4 gene that significantly alter its function are quite rare. This suggests that most of that huge variability we see isn't due to different forms of the enzyme itself, but likely due to genetic differences in the regulatory regions that control how much CYP3A4 protein gets made.
So differences in the on-off switch or the dimmer switch for the gene?
Kind of like that, yes, affecting the quantity, not necessarily the quality of the enzyme. CYP3A5, its close relative, is a bit different, though. It does have a very common variant with a major functional impact.
Okay, what's the story with CYP3A5?
There's a common SNP, designated CYP3A53, that creates a faulty splice site in the gene's RNA message. This leads to improper splicing and results in a non-functional protein or drastically reduced activity. Most people of European or Asian ancestry actually carry 2 copies of this 3 allele, meaning they have very little or no functional CYP3A5 enzyme. However, the original functional allele, often called 1, is much more common in individuals of African ancestry. So on average, people of African descent tend to have higher overall CYP3A activity because they're more likely to have functional CYP3A5 in addition to CYP3A4.
And the material links the CYP3A5 difference to specific drug outcomes.
It does. It mentions the immunosuppressant drug tacrolimus used to prevent organ transplant rejection. Tacrolimus is metabolized by both CYP3A4 and CYP3A5. Because individuals of African ancestry are more likely to express functional CYP3A5, they tend to clear tacrolimus more quickly.
Meaning they might need higher doses.
Exactly. They often require higher doses to achieve the same therapeutic drug levels and prevent rejection compared to individuals without functional CYP3A5. On the flip side, it mentions the chemotherapy drug vincristine. Higher CYP3A5 activity seems to be associated with a lower risk of developing vincristine-induced neuropathy or nerve damage, presumably because faster clearance leads to lower exposure to the toxic drug.
Okay, interesting trade-offs. What about CYP2D6, the poster child from Figure 68.1?
Right. CYP26 metabolizes the second largest number of drugs after the CYP3 family, and it is incredibly polymorphic, just loads of genetic variation affecting its function. The poor metabolizer, PM phenotype, with little to no activity is quite common in some groups, around 20% in Europeans, maybe 10% in Africans, but much rarer, only around 2% in Asians. And then you have the ultra-rapid metabolizers, UMs, due to gene duplications, who are found in various populations but seem particularly prevalent in parts of East Africa, the Middle East, and Oceania, sometimes reaching frequencies of 3% or more.
And the clinical consequences can be really severe, right? The codeine example is unforgettable.
It really is the classic cautionary tale. Codeine itself isn't a very effective pain reliever. It's a prodrug. It needs to be converted by CYP2D6 into morphine to provide significant analgesia.
So if you're a poor metabolizer,
If you're a PM, you have little or no CYP2D6 enzyme activity. You can't make much morphine from codeine, so you'll get little or no pain relief. The codeine just won't work well for you.
and if you're an ultra-rapid metabolizer,
That's where the danger lies. If you're a UM, you have excess CYP2D6 activity due to those extra gene copies. You convert codeine to morphine very, very rapidly and efficiently. This can lead to unexpectedly high, potentially toxic levels of morphine in your system, even from a standard dose of codeine.
and high morphine levels cause respiratory depression, slow or stopped breathing.
Exactly. The most serious risk is respiratory depression, which can be fatal. The source explicitly mentions tragic cases where children, who were later found to be CYP2D6 UMs, died from respiratory depression after receiving standard doses of codeine, often following tonsillectomy or adenoidectomy surgery.
Just awful, and that led to major warnings.
Yes. It led the FDA to issue its strongest warning, a black box warning, and ultimately to contraindicate codeine use entirely in children under 12 and recommend extreme caution in adolescents undergoing those specific surgeries. It's a direct, tragic link. Genotype-altered metabolism, severe adverse event.
A stark reminder of why this matters. What about other drugs affected by CYP2D6? Beta blockers were mentioned.
Yes, many commonly used beta blockers, metoprolol, carvedilol, propafenone, which also has antiarrhythmic effects, are primarily cleared by CYP2D6. In PM individuals, these drugs hang around much longer, leading to higher concentrations.
So exaggerated effects.
Potentially, yes. Increased risk of side effects related to excessive beta blockade, like significant slowing of the heart rate, bradycardia, or triggering bronchospasm, especially in patients with asthma or COPD. Even Timol eye drops for glaucoma can be absorbed systemically and cause these effects in PMs. The material also mentions tamoxifen, the breast cancer drug. It's also a prodrug activated by CYP2D6 to its more potent metabolite, endoxifen. So there's been a lot of research suggesting that lower CYP2D6 activity might lead to lower endoxifen levels and potentially reduce tamoxifen's effectiveness, although demonstrating this conclusively in large trials has been complex.
And it also mentions how some other drugs can interfere. The phenocopying thing again.
Right. Common antidepressants, specifically the SSRIs fluoxetine, Prozac, and paroxetine, Paxil, are potent inhibitors of the CYP2D6 enzyme. If you take one of these drugs, they can effectively shut down your CYP2D6 activity, even if genetically you're an EM. So you functionally behave like a PM for any other drugs that rely on CYP2D6. It mimics the genetic effect.
Fascinating. Drug interactions mimicking genetic traits. Okay, next up, CYP-C19. Another important one.
Very important. And like CYP2D6, it shows big differences in activity across populations. The PM phenotype for CYP-TC19 is quite common in Asians, around 20%, and extremely common in some populations in Oceania, over 50% in some studies, but it's much less frequent, only 2–3% in Europeans and Africans.
And the example given relates to treating stomach ulcers with PPIs.
Yes, the proton pump inhibitor omeprazole, Prilosec, it's mainly metabolized by CYP2C19. Studies found actually after the drug was widely used that ulcer cure rates with standard omeprazole doses were significantly lower in patients who were extensive metabolizers, EMs.
Because they cleared the drug too fast.
Exactly. Faster clearance meant lower drug exposure, less acid suppression, and lower cure rates. In contrast, PMs who cleared it slowly and had higher drug levels had much better cure rates. This was an early clinical hint that knowing CYP2C19 status could potentially guide dosing, maybe suggesting higher doses for EMs, though this wasn't part of the initial development or labeling.
And CYP2C19 is also critical for clopidogrel, the antiplatelet drug, Plavix.
Absolutely critical. Clopidogrel is another major prodrug. It needs to be activated by CYP2C19 to form its active metabolite that actually inhibits platelet aggregation. And numerous studies, both looking backward, retrospective, and tracking patients forward, perspective, have clearly shown that people carrying one or two reduced function alleles for CYP2C19 have decreased activation of clopidogrel.
Leading to less antiplatelet effect.
Yes, and consequently a higher risk of major adverse cardiovascular events like heart attacks, strokes, or clots forming inside coronary stents, especially in the period after stent placement. This is a very well-established association.
And again, the PPI interaction is mentioned.
Yes, the source reiterates that omeprazole and some other PPIs are inhibitors of CYP2C19. So taking a PPI concurrently with clopidogrel can further reduce its activation, potentially worsening outcomes, especially in someone who might already be an IM or PM genetically.
That interaction seems really important for patients on both drugs. Okay, how about CYP-TC9?
CYP-TC9 also has common variants, particularly the 2 and 3 alleles, that encode enzymes with significantly reduced catalytic function. These variants are linked to increased risks with several drugs. For instance, increased neurologic side effects with the anti-seizure drug phenytoin, and increased risk of hypoglycemia, dangerously low blood sugar, with the sulfonylurea diabetes drug glipizide, because both are cleared by CYP2C9. And most famously, CYP2C9 variants are a major factor in determining the required dose for warfarin.
Warfarin again. It seems like genetics is key for that drug.
Hugely key. It's notoriously difficult to dose because of its narrow therapeutic window and high inter-individual variability. The material states that polymorphisms in CYP2C9, primarily 2 and 3, reducing metabolism, and in the promoter of VKORC1, the drug's target affecting sensitivity, together can explain up to 50% of the variability in the stable warfarin dose needed between patients.
Half the variability explained by just those two genes.
Yeah.
That's massive.
It is. Patients who are homozygous for loss-of-function CYP2C9 alleles might need tiny doses of warfarin compared to someone with normal function. Other genes like CYP4F2 involved in vitamin K metabolism also play a role, but CYP2C9 and VKORC1 are the big ones for dose prediction.
And is there another prodrug example linked to CYP2C9?
Yes, losartan, an angiotensin receptor blocker, ARB, used for high blood pressure. It's partially activated by CYP2C9 to a more potent metabolite. So patients who are PMs for CYP2C9 or those taking strong CYP2C9 inhibitors might not get the full blood pressure lowering effect from losartan because they can't activate it efficiently.
Okay, we've covered the major CYPs. Moving beyond them, the source mentions DPYD. What's that?
DPYD stands for dihydropyrimidine dehydrogenase. This enzyme is crucial for breaking down a class of chemotherapy drugs called fluoropyrimidines. These include 5-fluorosyl, 5-FU, which is very widely used, especially for colorectal cancer, and as oral prodrugs like capecitabine and Tegafur.
So DPYD detoxifies these cancer drugs.
Essentially, yes, it's the first and rate-limiting step in their catabolism. Genetic variants that lead to reduced or completely absent DPYD enzyme activity are really dangerous here. If you have deficient DPYD activity, you can't break down these drugs properly. Standard doses can lead to a massive buildup of toxic drug levels, causing severe, potentially fatal toxicities like profound bone marrow suppression, wiping out blood cells, severe diarrhea and mucositis, inflammation of the gut lining, and neurotoxicity.
That sounds incredibly risky, so testing seems vital.
It's becoming increasingly recognized as vital before starting these therapies. The risk is particularly high for individuals homozygous for loss-of-function alleles, but even heterozygotes, intermediate metabolizers, are at significantly increased risk and require substantial dose reductions, according to guidelines. Testing for DPYD deficiency can literally be life-saving.
Wow. Okay, the source then groups some others as transferase variants, starting with TPMT.
Right, TPMT is thiopurine S-methyltransferase. This enzyme is responsible for inactivating another important class of drugs, the thiopurines. This includes 6-mercaptopurine, 6-MP, a cornerstone drug for treating childhood acute lymphoblastic leukemia, ALL, an immunosuppressant used widely for autoimmune diseases like Crohn's or rheumatoid arthritis, and also in organ transplantation.
And genetics matters here too.
Immensely. This is another high-impact example, similar to DPYD. About 1 in 300 individuals are homozygous for inactive TPMT alleles. They have virtually zero TPMT enzyme activity. If these individuals receive standard doses of 6-MP or azathioprine, they cannot metabolize the drug properly via this pathway. This leads to a shunt towards producing highly toxic metabolites that accumulate and cause severe life-threatening bone marrow suppression or myelosuppression. Complete aplasia is possible.
So another case where standard doses can be fatal for a genetically susceptible subgroup?
Exactly. Testing for TPMT status before starting thiopurines is now standard practice in many places. Conversely, individuals who are homozygous for the fully functional alleles might metabolize the drug very quickly, potentially leading to lower active metabolite levels and maybe reduced therapeutic effect, requiring higher doses. And interestingly, the source adds that more recent research identified variants in another gene, NUDT15, that also impact thiopurine metabolism. Loss-of-function variants in NUDT15, particularly common in East Asian populations, also lead to accumulation of toxic metabolites and increased risk of myelosuppression. So sometimes multiple genes contribute to the risk for the same drug.
Two different genetic pathways affecting risk for the same drug class. Okay, what about N-acetyltransferase, NAT?
NAT, specifically the NAT2 enzyme, is responsible for acetylation, another metabolic pathway. Genetic variations in NAT2 lead to distinct phenotypes. People are classified as either rapid acetylators or slow acetylators.
Slow versus rapid, how common is that?
The slow acetylator phenotype is actually quite common, around 50% frequency in European and African populations, though less common in East Asians.
And being a slow acetylator has clinical consequences.
Yes. Because they metabolize certain drugs more slowly via this pathway, slow acetylators have prolonged exposure to the parent drug. This increases their risk for specific adverse effects with certain medications. The examples given are an increased risk of drug-induced lupus syndrome with procainamide and hydralazine, and an increased risk of liver toxicity, hepatitis, with the tuberculosis drug isoniazid.
Okay. Another key enzyme mentioned is UGT1A1.
UGT1A1 is involved in glucuronidation, attaching a glucuronide sugar molecule to drugs or substances like bilirubin to help eliminate them. There's a very common variation in the promoter region of the UGT1A1 gene, often called the UGT1A128 allele, associated with Gilbert syndrome.
Gilbert syndrome, that's usually mild, isn't it?
Yeah.
Elevated bilirubin.
Usually, yes. People homozygous for this variant have reduced UGT1A1 transcription, less enzyme, and mildly elevated bilirubin, which is typically harmless. However, pharmacogenetically, it's important. These individuals with reduced UGT1A1 activity are at significantly higher risk of severe toxicity, especially severe diarrhea and bone marrow suppression when treated with the chemotherapy drug irinotecan.
Why irinotecan?
Irinotecan is a prodrug, and its active metabolite, SN38, is primarily detoxified and eliminated by UGT1A1-mediated glucuronidation. If you have low UGT1A1 activity due to the 28 allele, SN38 builds up to toxic levels.
And the source links UGT1A1 to an HIV drug too, atazanavir.
Yes. Interestingly, the HIV protease inhibitor, atazanavir, is itself an inhibitor of the UGT1A1 enzyme. So if a patient who already has reduced UGT1A1 activity due to the Gilbert's variant takes atazanavir, the drug further inhibits their remaining enzyme activity. This can cause their bilirubin levels to rise quite significantly. While this hyperbilirubinemia is generally considered benign, just cosmetic jaundice, not liver damage, the source notes it can be clinically confusing. It might be mistaken for drug-induced liver injury, leading to unnecessary worry, investigations, or even stopping an effective HIV regimen.
Good to know. Finally, in this kinetics section, the source talks about transporter variants. So not enzymes breaking drugs down, but proteins moving them around.
Exactly. Drug transporters are proteins embedded in cell membranes that act like pumps or channels, actively moving drugs and other substances into or out of cells, across tissues like the gut lining or the blood-brain barrier, or into organs like the liver and kidney for elimination. Genetic variations in these transporters can definitely affect drug response. A major one is SLCO1B1, which codes for the OATP1B1 uptake transporter in the liver. This transporter helps pull statins, like simvastatin, out of the blood and into the liver. Common variants in SLCO1B1 reduce the transporter's function. If the transporter isn't working well, less statin gets into the liver, meaning higher levels remain circulating in the blood and potentially getting into muscle tissue. This is strongly linked to an increased risk of statin-induced myopathy, muscle pain, weakness, and in rare cases, severe muscle breakdown.
So transport defect increases risk of that major statin side effect.
Correct, especially with simvastatin at higher doses. Another example is ABCB1, which codes for P-glycoprotein, PGP, an efflux transporter that pumps drugs out of cells. It's found in the gut, blood-brain barrier, kidney, and other places. Variants in ABCB1 may influence the absorption or distribution of drugs like digoxin, potentially affecting toxicity risk, although the clinical impact here has sometimes been harder to pin down consistently than for SLCO1B1. And variants in transporters like MADI1 and MADI2, involved in excreting drugs into urine, have been linked to variability in the glucose-lowering effect of the diabetes drug metformin. So yes, transporters are definitely another key piece of the pharmacokinetic puzzle influenced by genetics.
Okay, so we've covered how genetics affects what the body does to the drug pharmacokinetics. Now let's flip it. Pharmacodynamics, how the drug affects the body, its target interactions, the downstream response. Genetics matters here too.
Absolutely. Genetic variations can directly alter the drug's target, the receptor, enzyme, or ion channel it's designed to interact with, or they can affect other molecules in the biological pathway that the drug influences. The source brings back the VKORC1 example here, highlighting its dual role. We mentioned the promoter variant affects the amount of VKORC1 protein, kinetics-related effect on dose via target availability. But VKORC1 itself is the enzyme that warfarin inhibits, so the variant directly impacts the pharmacodynamic sensitivity to the drug by changing the level of the target.
It's a great illustration of how kinetics and dynamics can be intertwined through genetics. What about other pharmacodynamic examples? Beta receptors were mentioned again.
Yes. The beta-adrenergic receptors, which one found mainly in the heart, AGFU in the lungs, and other tissues, are targets for widely used drugs. Beta blockers for heart conditions, beta agonists for asthma. Variations in the genes coding for these receptors themselves, ADRB1 and ADRB2, have been studied extensively. Since the drug binds directly to the receptor, a change in the receptor's amino acid sequence due to a genetic variant could potentially alter how tightly the drug binds or how effectively it activates or blocks the receptor's signaling pathway.
Any specific examples linking beta receptor variants to clinical outcomes?
The material highlights one specific variant in the VAV1 adrenergic receptor gene, ADRB1, known as R389G, arginine versus glycine at position 389. In a clinical trial of an investigational beta blocker, busindolol, for heart failure, patients carrying the arginine allele, R389, seemed to have significantly better outcomes.
Better outcomes? How?
They showed decreased mortality and a lower incidence of atrial fibrillation compared to patients with the glycine allele, G389, when treated with busindolol. This suggests that this specific target variant influences the degree of clinical benefit derived from that particular beta blocker in heart failure patients. It's a direct pharmacodynamic effect on efficacy.
Interesting. Can genetics also make someone more susceptible to a drug triggering an underlying condition, even if the drug isn't directly targeting the gene for that condition, like unmasking a hidden problem?
Yes, definitely. Drugs can interact with latent genetic predispositions. The source mentions porphyrias, a group of rare metabolic disorders affecting heme synthesis. People with certain genetic forms of porphyria are susceptible to acute attacks, often involving severe neurological symptoms. Certain drugs, especially those known to induce CYP enzymes, can precipitate these attacks in genetically susceptible individuals.
And G6PD deficiency came up again. That seems like a classic example.
It is. Glucose-6-phosphate dehydrogenase G6PD deficiency is very common worldwide, especially in malaria endemic regions. G6PD enzyme protects red blood cells from oxidative stress. People with G6PD deficiency have red cells that are vulnerable to damage from certain oxidant substances, including some drugs. When they take these drugs, classic examples are the anti-malarial primaquine, certain sulfa drugs, and the uric acid-lowering drug resburicase, they can experience acute hemolytic anemia, sometimes severe, where their red blood cells rapidly break down. Rasburicase is highlighted as being safe in people with normal G6PD, but potentially causing severe hemolysis in deficient individuals.
Another clear case where pretesting is crucial to avoid harm. What about RYR1?
RYR1 codes for the ryanodane receptor type 1, a calcium channel in skeletal muscle. Certain mutations in RYR1 predispose individuals to malignant hyperthermia, MH. These individuals are typically perfectly healthy until they are exposed to specific triggering agents, usually volatile anesthetic gases like sevoflurane, desflurane, or the muscle relaxant succinylcholine often used during surgery. In susceptible individuals, these agents trigger uncontrolled calcium release from muscle stores, leading to a hypermetabolic crisis, muscle rigidity, skyrocketing body temperature, rapid heart rate, acidosis, which is rare but life-threatening if not treated immediately.
And long QT syndrome. That sounds like another hidden genetic risk interacting with drugs.
Exactly. Congenital long QT syndrome is a collection of genetic disorders affecting ion channels in the heart muscle, leading to a prolonged QT interval on the electrocardiogram and an increased risk of a dangerous arrhythmia called torsades de pointe, which can cause fainting or sudden death. Many drugs are known to prolong the QT interval as a potential side effect. While most people tolerate modest prolongation, in some individuals who experience severe QT prolongation or develop torsades de pointe when exposed to these drugs, it's subsequently discovered they had an underlying, perhaps previously undiagnosed, genetic long QT syndrome. The drug essentially unmasked their predisposition.
Okay, so genetics affects kinetics, dynamics, and even predisposes to certain drug-triggered conditions. What about the immune system's role? Can genetics influence allergic or hypersensitivity reactions to drugs?
Yes, this is another critical area where genetics, particularly variation in the human leukocyte antigen HLA system, plays a huge role. The HLA genes code for proteins on the surface of cells that are crucial for presenting antigens to the immune system, especially T cells. Specific HLA alleles have been found to be very strongly associated with the risk of developing severe, potentially life-threatening immune-mediated adverse drug reactions. The most dramatic examples are severe skin reactions like Stevens-Johnson syndrome, SJS, and toxic epidermal necrolysis, TEN, which involve widespread blistering and sloughing of the skin and mucous membranes.
And specific HLA types are linked to specific drugs causing these reactions. Table 68.2 lists some.
Precisely. These are some of the strongest and most clinically important pharmacogenetic associations known. For example, the HLA-B 15.02 allele confers a very high risk of SJS/TEN, specifically with the anticonvulsant drug carbamazepine, and also phenytoin, primarily in individuals of Asian descent, where this allele is more common. Another allele, HLA-B 31.01, is associated with SJS/TEN and also milder skin reactions to carbamazepine in both Caucasian and Asian populations. And HLA-B 57.01 is famously linked to a severe multi-organ hypersensitivity reaction, including risk of SJS/TEN with the HIV drug abacavir. This same allele, HLA-B 57.01, is also associated with liver injury, hepatotoxicity, caused by the antibiotic flucloxacillin.
So carrying that specific HLA marker puts you at massively increased risk if you encounter that particular drug.
That's the interpretation, yes. The association, particularly for HLA-B 57.01 and abacavir, and HLA-B 15.02 and carbamazepine in Asians is extremely strong. And again, the frequencies of these risk alleles vary significantly across different ancestral populations, meaning the baseline risk for these specific severe reactions differs between groups. The source also mentions that other immune reactions, like drug-induced liver injury more generally, and DDRES syndrome, drug rash with eosinophilia and systemic symptoms, have also been linked to HLA variants.
Is having the HLA risk allele enough on its own to cause the reaction? Like if you have HLA-B 57.01 and take abacavir, are you guaranteed to react?
That's a really interesting point the source touches on. It suggests that while the specific HLA allele seems necessary for the reaction, or at least for the high risk, it's often not sufficient on its own. It uses the HLA BP7.01 example. About 55% of carriers who take abacavir will develop the hypersensitivity reaction. That's high, but not 100%. But for flucloxacillin-induced liver injury, also linked to HLA-B 57.01, the reaction rate is tiny, maybe 1 in 10,000 carriers. Same HLA allele, but vastly different reaction rates for the two drugs.
Exactly. This strongly implies that there must be other factors involved, maybe related to the drug itself, how it's processed, or some other aspect of the individual's immune system or environment. A third factor, as the source puts it, that's needed along with the HLA allele to actually trigger the full-blown reaction. What that third factor is often not yet clear.
This next section shifts focus again, which is really interesting. It's not just our human genome, but sometimes the genome of the thing we're trying to treat, like a tumor or an infectious agent that determines if a drug works.
Precisely. When we use antimicrobial drugs or anti-cancer drugs, the drug's target is often a component of the pathogen or the cancer cell itself. So genetic variations or mutations within the genome of that tumor or microbe can be the critical factor determining drug efficacy or resistance.
Let's start with cancer. Tumor genotyping, sequencing the cancer's DNA, is becoming much more common, right? Yes, it's rapidly becoming standard practice for many cancer types. The idea is to identify the specific driver mutations or genetic alterations within the tumor cells that are making them grow uncontrollably. Many newer cancer therapies, the so-called targeted therapies, are designed to specifically attack cells carrying those particular alterations.
So you need to know if the tumor has the target before you use the drug designed for that target.
Exactly. It's essential for selecting patients who are likely to benefit. The material gives several key examples. Trastuzumab, Herceptin, and Lapatinib work for breast cancer, but only if the tumor cells are overexpressing the HER2/neu receptor protein due to gene amplification. If the tumor is HER2 negative, these drugs won't work. Imatinib, Gleevec, revolutionized treatment for chronic myelogenous leukemia, CML, because it targets the specific BCR-ABL1 fusion kinase produced by the Philadelphia chromosome translocation found in CML cells. No Philadelphia chromosome, no BCR-ABL1, no benefit from imatinib for CML. Imatinib also hits the KAKIT kinase, making it effective for gastrointestinal stromal tumors, GIST, driven by APIC mutations.
More examples. This really highlights the precision aspect.
Yes, vemurafenib targets melanoma, but specifically melanomas with the V600E mutation in the BRAF gene. It doesn't work against melanomas with normal BRAF. Crizotinib is highly effective, but only in non-small cell lung cancers that have specific mutations or rearrangements involving the ALK gene. And the EGFR inhibitors panitumumab and cetuximab, used in colorectal cancer, lose efficacy if the tumor has mutations in the KRAS gene downstream, because the mutation bypasses the need for EGFR signaling.
So for all these targeted drugs, testing the tumor's genetics is basically required to guide treatment.
Absolutely, it determines eligibility, but the source also points out the major challenge in targeted cancer therapy: acquired resistance. Cancer cells are genetically unstable and can evolve. Even if a drug works brilliantly at first by hitting a specific target, the tumor can often find ways around it by developing new mutations or activating alternative pathways. The vemurafenib example for BRAF mutant melanoma is mentioned again. Initial responses are often dramatic, but resistance usually develops relatively quickly when it's used alone. That's why combination therapies, like adding a MEK inhibitor to block a downstream pathway, are now standard practice, trying to prevent or delay resistance.
And what about immune checkpoint inhibitors, the ones that unleash the patient's immune system against the cancer? Genetics plays a role there, too.
Well, the source notes the variability. These drugs can lead to amazing, long-lasting remissions in some patients, which is fantastic, but others don't respond well, and a significant fraction experience serious immune-related side effects, colitis, pneumonitis, myocarditis, etc., where the revved-up immune system attacks normal tissues. Understanding why there's such variability in both benefit and harm is a huge area of research. It likely involves a complex interplay between the patient's own immune system genetics, like HLA types, the genetic characteristics of the tumor itself, like its mutation burden or expression of immune markers like PD-L1, and potentially the gut microbiome. The source doesn't detail specific patient genetic factors here, but highlights the variability as a major challenge needing explanation.
Okay. And what about infectious agents? How does their genetics affect drug treatment?
Pathogens evolve resistance through genetic changes too. Bacteria develop antibiotic resistance via mutations or acquiring resistance genes. Viruses mutate constantly. The material gives one specific pharmacogenetic example related to HIV. The HIV entry inhibitor drug Maraviroc works by blocking a human protein called CCR5, which some strains of HIV use as a co-receptor to get inside immune cells. However, other HIV strains use a different co-receptor called CXCR4 or can use either, dual tropic. Maraviroc only works against HIV strains that rely solely on CCR5, so-called R5 tropic virus.
So you have to test the virus first.
Exactly. Before prescribing Maraviroc, a viral tropism test is needed to determine if the patient's specific HIV strain uses CCR5. If it uses CXCR4, Maraviroc will be ineffective. So here, it's the virus's characteristic determined indirectly by its genetics that guides the therapy choice.
Wow. Okay, we've covered genetics affecting our body's handling of drugs, the drug's effect, immune reactions, and even the genetics of tumors and viruses. This is incredibly complex, but also holds so much promise. How is this knowledge actually starting to filter down into everyday clinical practice? Are doctors using this routinely?
That's the crucial transition point from discovery to application. And it's happening, but maybe more slowly and unevenly than some might have hoped. The discovery of all these variants with significant clinical effects obviously raises the exciting possibility of using this information proactively to guide treatment. The goal, as we've discussed, is better drug selection, optimized dosing from the start, and critically, avoiding drugs predicted to cause severe harm or be ineffective for that specific patient based on their genetic profile, moving beyond trial and error.
So getting it right, or at least righter the first time.
That's the ambition. And the FDA is definitely incorporating more pharmacogenetic information into drug labels, which helps raise awareness. But deciding when it's appropriate and practical to actually do the genetic testing in a clinical setting depends on a bunch of factors. The source highlights. How big is the genetic effect? Is it a minor tweak or a major risk? How strong is the evidence linking the variant to the outcome? Is it just an association or proven in rigorous trials? Do we understand the biology well? And then there are practicalities. Is the test reliable and available? How quickly can you get results? And importantly, what's the cost versus the expected benefit?
Right, lots of hurdles. The material mentions two main strategies being looked at for implementation: point of care versus preemptive testing. Can you explain those?
Sure. Point of care testing is reactive. A doctor decides to prescribe a specific drug known to have a critical pharmacogenetic link, like abacavir or carbamazepine. At that moment, they order a rapid genetic test just for the relevant variants for that drug. The key is getting the result back very quickly, ideally within hours, maybe a day, so it can directly inform that prescribing decision right then and there.
So, test for the drug you're about to prescribe. Reactive. What's preemptive?
Preemptive is proactive and much broader. The idea is to test a patient, perhaps just once in their life, for a large panel of many important pharmacogenetic variants relevant to dozens or even hundreds of different drugs across many therapeutic areas. This testing happens before any specific need arises. The results are then stored securely, usually integrated into the patient's electronic health record, EHR. Then, anytime in the future, if a doctor goes to prescribe any drug covered by the panel, the EHR system, using clinical decision support, CDS tools, can automatically check the patient's stored genetic data.
And alert the doctor.
Exactly. It can fire off an alert saying, "Warning, this patient's genotype suggests increased risk of toxicity with this drug," or reduced efficacy predicted, and ideally offer specific recommendations, like suggesting an alternative drug or modified dose, often linking directly to established guidelines like those from CPIC.
Preemptive sounds ideal in theory, have all the info ready whenever needed, but implementing either of these must have challenges.
Huge challenges. While getting reliable genetic testing done, assay validity, is less of an issue now, integrating the results effectively into the clinical workflow is tough. Getting the data into the EHR in a standardized way, building reliable and user-friendly CDS systems that doctors will actually use and trust, that requires massive effort, standardization, and ongoing work by national consortia. The source also points out another complexity. We know a lot about the common variants listed in tables like 68.1. But with widespread sequencing, we're finding tons of rare variants in these same genes. What do they do? Often we have no idea about their functional impact, making it really hard to give clinical advice based on them. Predicting function for novel variants is a major bioinformatics challenge.
And figuring out the precise dose adjustment, even for known variants, isn't simple, is it?
Not at all. It depends on whether you have one variant allele or two, whether the variant causes reduced function, complete loss of function, or even increased function, like alleles. And how do you combine the effects if someone has variants in multiple genes affecting the same drug, like CYP2C9 and VKRRC1 for warfarin? This is where expert groups like CPIC, Clinical Pharmacogenetics Implementation Consortium, and the DPWG, Dutch Pharmacogenetics Working Group, are invaluable. They systematically review the evidence and develop peer-reviewed guidelines for specific drug-gene pairs, suggesting concrete actions based on genotype. But as the source notes, these guidelines tell you what to do if you have the genotype. They generally don't dictate when or if you should order the test in the first place. That implementation decision often falls to healthcare systems or individual clinicians.
Which brings us back to that key question. Does doing the testing actually improve patient outcomes compared to not testing? What's the evidence?
This is where the debate often lies. One viewpoint, as mentioned before, is that for variants with large, well-understood effects like TPMT and thiopurines or DPYD and 5-FU, the physiological consequence is so clear that requiring large RCTs to prove testing helps might be overkill, maybe even unethical. It's like adjusting kidney drug doses for kidney failure. We do it based on physiology.
If the enzyme is missing, the drug will build up. That's clear.
Right. The counter-argument is that for many other gene-drug pairs, the genetic effect might be smaller or interact complexly with other factors. In these cases, proponents argue you really need high-quality evidence, ideally from RCTs, showing that using the test actually leads to demonstrably better clinical outcomes, fewer adverse events, better disease control, lower healthcare costs to justify widespread implementation.
So what has the evidence shown from trials and real-world implementations? Is it clear-cut?
It's definitely mixed. The standout success story is HLA B57.01 testing for abacavir. An RCT definitively showed that screening and avoiding abacavir in carriers virtually eliminated the severe hypersensitivity reactions. That's a clear win for testing.
What about the carbamazepine example with HLA-B 15.02? Testing was mandated in some places. Did it help?
The outcome there was more complex, according to the source. While the intent was good, in some regions where testing is mandated but perhaps not easily accessible or integrated, prescribers reportedly just stopped using carbamazepine altogether. They switched to alternatives like phenytoin.
Which also carries SJS10 risk, sometimes linked to the same HLA allele.
Exactly. So the overall rate of severe skin reactions didn't necessarily decrease. It shows that implementation isn't just about having a test, it's about the entire system's response, including education, access, and appropriate alternative choices.
And the warfarin trials. That seemed like a prime candidate for genetic guidance.
Yes, huge effort went into warfarin pharmacogenetic trials. Early large RCTs comparing genotype-guided dosing using CYP2C9V KORC1 versus standard clinical dosing often showed disappointing results, maybe a slight improvement in time spent in the therapeutic range early on, but often no significant difference in major clinical outcomes like bleeding or clotting. However, the source does mention a more recent trial that did show genotype guidance significantly reduced the frequency of over-anticoagulation, very high INR values, early in therapy. Since over-anticoagulation is the main driver of bleeding risk, this is potentially clinically important. Retrospective studies also link specific variants, like CYP2C93, CYP4F2, to bleeding risk. So the picture's nuanced. Maybe testing helps reduce bleeding risk more than preventing clots.
And clopidogrel with CYP2C19.
There, some interesting trials took a different approach. They compared newer antiplatelet drugs, ticagrelor, prasugrel, against clopidogrel, but specifically enrolled patients without the CYP2C19 loss-of-function variants, the ones expected to metabolize clopidogrel normally. In one such trial, clopidogrel actually performed better than the newer agent in this genetically selected group. In another, there was a trend suggesting clopidogrel might be better, though it didn't hit the study's main target. This hints that maybe clopidogrel is a very good drug if you can activate it properly, which leads back to that strategy the source mentions. Maybe the best use of testing for drugs like warfarin and clopidogrel, where effective but genetically variable drugs compete with newer, more expensive, less variable alternatives, is to identify the large group of patients who don't have the problematic variants. They could potentially stick with the older, cheaper drug quite safely and effectively, reserving the newer agents for those identified by genetics as being poor candidates for the standard therapy.
That makes a lot of practical sense, using genetics to optimize existing therapies, not just jump to new ones. Lastly, let's talk about drug development itself. How is all this genetic knowledge changing how new drugs are discovered and brought to market?
It's having a pretty profound impact, actually. Genetic tools and insights are being used much earlier in the pipeline now, primarily to help identify and, crucially, validate potential drug targets. The thinking is, if human genetic evidence already links variation in a particular gene or pathway to the disease you want to treat, then developing a drug to modulate that target has a much higher chance of success. It's like getting a strong hint from human biology itself that hitting this target will matter. Conversely, genetic data might flag potential safety issues early on if variants suggest problems with metabolism or off-target effects.
So genetics helps pick more promising targets and potentially avoid costly failures later on.
Exactly, it de-risks the process somewhat. Genetics is also fueling drug repurposing efforts. By analyzing links between genetic variants associated with certain diseases and the known targets of existing drugs in large population databases, like those linked to EHRs, researchers can spot potential new uses for old drugs. The rheumatoid arthritis example in the text illustrates this. GenoA studies identified genes linked to RA risk. Some of the proteins coded by these genes are already targeted by drugs approved for other conditions, like cancer or other autoimmune diseases. This genetic link provides a rationale to test those existing drugs for efficacy in RA.
That could be much faster and cheaper than starting from scratch with a totally new compound. And the source gives that really powerful PCSK9 example where finding a protective genetic variant led directly to a new class of drugs.
Yes, the PCSK9 story is a landmark case study. It started with finding rare gain-of-function mutations in the PCSK9 gene that caused extremely high cholesterol, familial hypercholesterolemia. That was interesting, but the breakthrough came when researchers looked the other way. They searched large population studies and found individuals with rare loss-of-function variants in PCSK9 variants that basically knocked out the gene's function. These individuals had significantly lower LDL cholesterol levels throughout their lives.
Naturally low cholesterol due to this gene variant.
Yes. And crucially, these individuals also had a substantially lower risk of developing coronary artery disease, fewer heart attacks. And importantly, they didn't seem to suffer any obvious ill effects from lacking PCSK9 function.
So nature's experiment showed that blocking PCSK9 is likely safe and effective for lowering cholesterol and preventing heart disease in humans.
Precisely. This powerful human genetic validation provided enormous confidence in PCSK9 as a drug target. It triggered a race to develop PCSK9 inhibitors, mostly monoclonal antibodies. And these drugs went from genetic insight to market approval in remarkably rapid time, less than 10 years, which is incredibly fast in drug development. It really showcased the power of human genetics for target validation.
And finding these rare protective variants requires massive amounts of data, right?
It does. You need very large population cohorts, often hundreds of thousands or even millions of individuals, with both genetic data from sequencing or genotyping and linked health information from EHRs, biobanks like the UK Biobank, or large epidemiological studies. Only with those huge numbers can you reliably detect these rare variants and assess their impact on health outcomes. The source also mentions similar stories for APOC3 variants protecting against high triglycerides and TRIB1 variants influencing lipids. The field is also moving beyond just single variants, whether rare or common. There's growing interest in using polygenic risk scores. These scores combine the small effects of potentially millions of common variants across the genome to get a more comprehensive estimate of an individual's genetic predisposition to a disease or potentially their likely response to a drug.
So moving from single genes to a whole genome picture of risk or response.
Kind of, yes. It's part of this broader trend towards systems pharmacology or systems medicine, trying to integrate multiple layers of biological data, genomics, transcriptomics, proteomics, metabolomics, with clinical data to understand complex diseases and drug responses from a more holistic perspective, aiming for more precise predictions of efficacy and toxicity.
Wow. Okay, that was a genuinely deep dive into this chapter on pharmacogenomics. We've gone from basic principles to specific enzymes, immune reactions, tumor genetics, clinical implementation, and drug development, just to try and synthesize some of the absolute key takeaways from the source material.
I think one core message is that important genetic variants influencing how drugs work are often quite common. They're not just rare oddities. And critically, their frequencies can differ dramatically across ancestral populations, which has major implications for global health equity.
And a primary way these variants exert their influence is by altering pharmacokinetics, changing how the body absorbs, distributes, metabolizes, or excretes the drug, leading to different drug concentrations.
Right. Variations in metabolic enzymes, especially the CYPs, and drug transporters are absolutely central to many clinically relevant pharmacogenetic effects. That's where a lot of the focus has been.
And given the sheer volume of information, all the genes, all the variants, all the drugs, it's crystal clear that no single doctor can possibly remember all of this or keep up with it manually.
Absolutely impossible. That's why leveraging technology is essential. The future really depends on effectively integrating individual pharmacogenetic data into smart electronic health record systems equipped with robust clinical decision support to provide actionable guidance at the point of care.
And finally, using genetics not just reactively but proactively, early in the drug development process, holds real promise for identifying better targets and developing safer, more effective, and more targeted therapies, potentially much faster. The PCSK9 example really drives that home. This exploration of pharmacogenomics really underscores just how unique we all are, biologically speaking, right down to the molecular level of how our bodies interact with medicines. It feels like a fundamental step towards truly personalized healthcare.
It absolutely is. Understanding these genetic underpinnings helps explain that variability we always see in medicine, why the same pill can be a lifesaver for one person, cause devastating side effects in another, and do nothing at all for a third. It provides a powerful framework for understanding and predicting individual differences.
And it's clearly not a static field. It's evolving incredibly quickly, with huge potential still untapped, both for using our current drugs better and for discovering entirely new therapeutic approaches.
Definitely. And it's such a great example of how progress in medicine increasingly relies on integrating knowledge from very different disciplines: molecular genetics, clinical pharmacology, bioinformatics, population health, data science. They all have to come together.
So for you listening, having absorbed all this detail about how your genes can influence your response to medicine, here's something to perhaps mull over. As genetic testing becomes cheaper, faster, and more accessible, potentially making your own detailed pharmacogenomic profile readily available to you, how might knowing that information fundamentally change how you think about your own health, how you interact with doctors, and the decisions you make about medications throughout your life? Thank you for joining us for this deep dive.
It's always a pleasure to really dig into these fascinating and important topics.